thank you all for being here today virtually. I appreciate, that everybody's here to hear about my work. I wanted to start out with a little bit of information about hosts and parasitoids, which are the types of species that I model in my work. I know from giving previous presentations that at this point, I tend to have a number of audience members with some horrified expressions on their faces. So I'm imagining some of your faces grimacing a little bit here. Parasitoids are species that lay their eggs on or inside the body of the host, ultimately killing that host, and then that provides the sustenance and the resources necessary for the parasitoid offspring to survive and develop. So in this picture here, we have some parasitoid larvae emerging from the body of a caterpillar host. Here we have some parasitoid eggs laid on the body of this host caterpillar. And over here, you can actually see the adult parasitoid, typically a wasp species, laying its eggs inside this pupal stage of its host. So Mark alluded to this a little bit, but we are interested in hosts and parasitoid dynamics, partially because parasitoids are incredibly abundant in the world. Estimates say that somewhere between 4 and 10% of all multicellular species in the world are actually parasitoids. And so they're very prevalent and they play an important ecological role in regulating the populations of their host species, which are oftentimes can be agricultural pests. And so there's interest in understanding how this interaction, how these two species interact, and can we predict the types of interactions there. In terms of a mathematical framework, hosts and parasitoids fall into a category of species that have discrete non-overlapping generations. That means that models that have continuous time that are maybe more appropriate for modeling populations of say people or other types of animals that have continuous birth and death rates. Those types of models aren't appropriate when we're modeling insects or annual plants that have this discrete life cycle. They have well-synchronized sequential stages. And so the mathematical framework that we'll be using is a difference equation models, where we have the host population here at time t+1 is some function of the host and parasitoid populations at the previous time step. And the parasitoid population at time t+1 is some function of the host and parasitoid populations at time t. So instead of getting a continuous population at any particular time, we get these discrete points, maybe on a yearly cycle of what is the population at that particular point in time. One of the motivating features in my work has been historically a lack of biological specificity in the timing of the different phases in the life cycle of the host. There's a model that's kind of been used for decades that doesn't necessarily accurately represent the biology of these systems. And so in formulating the models that I have analyzed, I chose to look at including biological specificity to say that I'm specifically writing down models that have density-dependent competition occurring prior to parasitism in the life cycle of the host species. So one kind of life cycle illustration that that matches that assumption is that our adults lay eggs. Those eggs hatch into larvae. The larvae is when the density-dependent competition primarily takes place, as they're eating leaves and munching down, trying to get nutrients. And then for certain species, parasitism might occur just in the later stages of the larval stage. And so the density-dependent competition is prior to the parasitism. After that parasitism, the survivors become pupae and adults, and that continues. Another scenario that this mathematical framework is going to describe also is the case of a pupal parasitoid. So instead of attacking larval stages, the parasitoid attacks the pupal stages. So again, the density-dependent competition is occurring prior to the parasitism in the life cycle of the host. And so that sets up the mathematical framework for the types of models that I analyzed throughout. My goal in this project, as Mark kind of alluded to, originally started with an interest in looking at the impacts of climate change on this biotic interaction of host and parasitoid. But because of some historical models that didn't necessarily have biological accuracy or specificity, I ended up spending some time starting off with difference equation models and analyzing the impacts of some specific biological assumptions on the population dynamics in these different possible models that you can write down without including space. And so specifically, I'll be looking at scramble versus contest competition in the host species and random versus parasitoid aggregation in the parasitoid species. Once I got a handle on that, those non-spatial problems, I was able to move to the IDE models and analyze the biotic interactions between the host and parasitoid and look at species survival in that scenario, as well as whether it's necessary to implicitly or explicitly represent space and what you can learn from those two different frameworks. So that's kind of a picture of where we're going in the narrative today. So part one are the difference equation models. The biological motivation here, the guiding questions. In general, populations of insects may have three typical patterns that you'll see. You'll either see erratic fluctuations, or a population might be maintained at a relatively low level, which would be certainly what farmers might hope, that their agricultural pests have low levels of population size. Or you might see cyclical populations, repeated growth and decline, outbreaks of these insects. So one of the, again, biological motivations here is that of biological control, which is defined as the control of some pest by an introduction of a natural enemy or predator. So the guiding question here is it possible to predict the effect of the introduction of a natural enemy, the parasitoid, on the host or pest population? So the components of the model that I have are the hosts at generation T, the female parasitoids at generation T. K is the carrying capacity of the hosts, kind of the maximum value of hosts that you'd expect to see if in the absence of the parasitoid. R0 is the net reproductive rate of hosts. Somewhat for historical reasons, some of the models actually use little r, which is the intrinsic rate of growth, but it's related to capital R. So you'll see both of those letters appearing today. Alpha is the searching efficiency of the parasitoids, and C is the parasitoid clutch size, the average number of eggs that are laid that then hatch and become adult, successful reproductive adult parasitoids. Mathematically, the natural next step, once we've kind of established our model, is to non-dimensionalize, simplify, remove some of the parameters. So X will scale the hosts by their carrying capacity. So when X equals 1, that means that the host species is at its carrying capacity. We'll scale the parasitoid population by the searching efficiency. And so that one doesn't have quite as natural of an interpretation as X equals 1 being the carrying capacity, but we'll see X and Y after we non-dimensionalize. In specifying that density-dependent competition occurs first in the life cycle, this is the set of equations that result. So what we see is that the The hosts at generation T undergo some density-dependent effects as they compete for resources, and then some percentage of that x g of x, some percentage of that is parasitized. Those that escape parasitism, this proportion here, become the adult, become the hosts at the next generation. Down here, those hosts that are parasitized give rise to some number of parasitoids at the next generation, and so that's our parasitoid equation there. So now we're going to discuss the specific model pieces that I'm using. As I mentioned earlier, one of the types of differences that you see in populations is the type of competition. So this is specifically, in this case, talking about the host species. So I'll be modeling both fractional per capita recruitment in models 1 and 3, which corresponds ecologically to contest competition, where there's essentially a winner in the competition for resources, and resources are not distributed equally. Some win and some lose. On the other hand, in our exponential per capita recruitment, which I'll be using in models 2 and 4, this is the ecological scenario of scramble competition, where resources end up distributed equally among the competitors. So, and mathematically, that's an exponential form that gives that type of behavior. I'm also going to be looking at a fractional and an exponential form of parasitism. So fractional parasitism will be used in models 1 and 2. And this is the case where parasitoids aggregate as they're searching for their hosts. The exponential parasitism in models 3 and 4 corresponds to parasitoids randomly searching for hosts. So I'll be looking at the impacts of those two different ecological scenarios. So ultimately, those different pieces are going to give us 4 models to analyze. Model 1 is kind of a base model where we choose the simplest form of recruitment, simplest form of parasitism, and it's frankly a little bit boring in terms of dynamics, but it's an important base case. And then I'll be considering what happens when I look at exponential recruitment and make that change to the base case, and what happens if I instead add exponential parasitism, and then what type of dynamics, what type of population dynamics do I see when I have both exponential recruitment and parasitism. So in analysis of these 4 models, each of them has these 3 equilibria, these 3 steady states to the system. The first is extinction. If there's no hosts and parasitoids, there will continue to be no hosts and parasitoids. The second steady state is where the host is at its carrying capacity and there are no parasitoids in the system. And the third is that where we have both species coexisting at some positive values. For models 1 and 2, I can explicitly solve for this x and y coordinate. But for models 3 and 4, because of the exponential functions, I'm unable to actually write these out explicitly, but can have an implicit representation of those. Analysis of these models proceeds by writing down the Jacobian matrix of the system, evaluating at the equilibrium, and looking at the eigenvalues of that Jacobian matrix. Ultimately, a particular equilibrium will be stable if the magnitude of the eigenvalues is less than 1 in magnitude. But practically speaking, I use a set of conditions known as the jury conditions to determine the stability. Each of these 3 jury conditions corresponds to a different type of bifurcation or a different type of change in the system if you violate that jury condition. So I won't go into the details here, but that idea will come up as we continue. So what do these different models tell us about the dynamics? In our first model, for the 2 biological parameters that we have here, if the biological parameters are anywhere in this gray region extending in both directions here, we know that the coexistence equilibrium is stable and that the system will go to that coexistence equilibrium if both R naught and B are greater than 1. When we add when we, instead of using the fractional recruitment, we use fractional recruitment, we use exponential recruitment, I get the jury 2 condition is part of the stability region here. And so if this condition is violated, the coexistence equilibrium is not stable. If I instead use exponential parasitism, I don't see the jury 2 condition having a role, but the jury 3 condition does decrease the size of the stability region and gives us an additional condition to satisfy with these parameters here. And if we cross this, if our parameters change such that this condition is violated, we lose stability. In the 4th model, where I have both exponential recruitment and exponential parasitism, the jury 2 and the jury 3 conditions show up as important conditions that define the stability region in our parameter space. And so we can see bifurcations resulting from both violating jury 2 and jury 3 conditions showing up in the potential dynamics of the system. So let's take a little bit closer look at model 2, which is the exponential recruitment with the fractional parasitism. So if we go back to our stability region here, I'm going to show you on the next slide in the XY, the host-parasitoid space, what the attractors in the system are if I have this particular green dot, which is just inside the stability region, if I have those biological parameters, and then what happens when I violate the second jury condition, what type of dynamics I get in that case when the parameters are just outside. So here we have the hosts on this axis, the parasitoids on this axis, and I'm showing you the stable attractors in the system. The red dot here in our first case corresponds to the coexistence equilibrium. So if our population starts somewhere near this coexistence equilibrium, it will eventually asymptotically approach this value. But there's also another stable attractor in the system. We see a 2 cycle where the population is bouncing back and forth between these 2 values. And the thing I want to emphasize here is that the hosts are, as we're oscillating back and forth, is both above and below the carrying capacity. So a pretty broad range here as it's oscillating. And so depending on where the system starts, you'll either see the coexistence behavior or you'll see this oscillation. For the parameter values that are just outside the coexistence equilibrium stability region, we'll see that the equilibrium is no longer stable, so it's not a stable attractor. And instead, I have a 4 cycle here where the population is going to be oscillating between these 4 different points. Notice that the parasitoid value of this is decreased as we've moved from this scenario to this scenario. So very low parasitoid values, and the host is oscillating both above and below the carrying capacity. So I want to look a little bit more in detail as what that transition looks like when we actually cross the boundary. So I'll show you in just a moment the dynamics and the bifurcation diagram as we cross this stability boundary at this red arrow location. Let's see. There we go. Okay. So in this slide, I'm showing the host population on this axis and what happens with a fixed B and a varying little r here. There's a lot going on in this diagram, so I want to emphasize a few things. These solid lines correspond to stable attractors in the system, and the dotted lines correspond to unstable phenomena. So at low values of the of R, there's a stable coexistence equilibrium, and then there's this 2 cycle that's unstable. But what you'll actually see in the system is a stable equilibrium point. For these sort of intermediate values of R, there's a 2-cycle above and below the carrying capacity for the hosts, with that coexistence equilibrium also a possibility depending on the initial condition of the system. And then as you increase R, you can see that there's the case of a 4-cycle that we saw in the previous slide. As R continues to increase, the, not shown here, but the parasitoid population continues to decrease until once we cross into this gray region, the parasitoid population has gone extinct, and so we just see the host population, 4 cycles, period doubling into 8 cycles, and ultimately into a chaotic regime. So we're seeing these relatively low period, high amplitude cycles. So if we compare the base case, model 1 with model 2, we see that when we add exponential recruitment, when the system has scramble competition, ecologically, that leads to the possibility of short period, high amplitude population cycles. And mathematically, that means that we see subcritical or supercritical period doubling or flip bifurcation showing up in the dynamics. On the other side of things, if we go back to using fractional recruitment and instead add exponential parasitism, we're going to see some different dynamics. So this is the stability region that I previously showed, zoomed in a little bit differently here, but this is the stability region for coexistence for model 3. And in the next slide, I'm going to show you the attractors for each of these green dots, each of these different sets of biological parameters in the system. So the first one is inside the stability region, and every point thereafter is outside the stability region. So here we have hosts on this axis and parasitoids on this axis again. This dot right here corresponds to that first green dot I showed you on the previous slide. This is the coexistence equilibrium that's stable for the original set of biological parameters. As we cross the stability boundary for this scenario, we see the development of tiny circles. They're called invariant circles, though their shape is not truly circular. And those invariant circles continue to grow as we increase the value of B. I want to note that this isn't like smoothly, the population isn't smoothly tracing out these curves. Instead, we're seeing a dense set of discrete points over time. You would see it bouncing around and eventually tracing out the curve densely, but not moving around it. Additionally, at some points for certain values of our parameters, you'll see instead of a invariant circle, there's a discrete set of points much higher than the 2 cycles and 4 cycles that we previously saw. Depending on the value, there's quite a number of points. And I'll note here that the parasitoid population you can see in each of these, as we increase B, is getting closer and closer to the axis, which is much more visible in our log scale plot here. You can see the parasitoid population is dropping 10 to the negative 10. So ecologically, if we had biological parameters in these ranges, we would expect the parasitoid population to be knocked extinct by some sort of environmental stochasticity, some sort of event that would knock out that population. But, looking at from another perspective, if we look at a bifurcation diagram, we have hosts on this axis and our B or our clutch size parameter on this axis. As we increase B for a fixed R, we see that we start out with the x coordinate of the coexistence equilibrium is stable, and it's decreasing in size. This is where what's called a Niemark-Sacker bifurcation occurs, which is that transition from the equilibrium to the invariant circles. Our invariant circles are growing in size, and then this is where the phase locking happens, which I'll go to the next slide here, and you can see these windows of phase locking. These thick regions correspond to invariant circles. These correspond to phase locking, where we can see a set of discrete points rather than kind of that traced out invariant circle. So All of the host values here are remaining below the carrying capacity though. So again, different from our second model. So kind of to sum that up, when we add the exponential parasitism or the random parasitoid searching to the model, ecologically, we're seeing these higher period, low amplitude population cycles as a possibility for the dynamics. And mathematically, we're seeing Neimark-Stacker bifurcations and invariant circles. The last question in this part then is what happens when we have both exponential recruitment and exponential parasitism in the system. And so if you recall, the stability region has both the jury 2 and jury 3 conditions showing up in the stability region, meaning that if we cross either of these curves, we're going to see the characteristic behavior of violating those jury conditions. So I won't go into details here, but just to summarize the results, when we have exponential recruitment and exponential parasitism, ecologically, we only see the higher period, low amplitude population cycles. We don't actually see those 2 cycles or 4 cycles. But mathematically, we do see both the subcritical flip bifurcation and the Neimark-Stacker bifurcation, with the invariant circles showing up as attractors in the system. To summarize the work in this project, I've analyzed some basic models of hosts and parasitoids under the assumption that density dependence precedes parasitism in the life cycle of the host. These models had not been previously analyzed or compared in the way that I approached the problem, and so I was able to characterize them and demonstrate that stronger nonlinearity in the recruitment function, that exponential form, gave rise to period doubling bifurcations, those flip bifurcations by another name, and stronger parasitism, the exponential form there, gave rise to Neimark-Stacker bifurcations. I'll also very briefly mention that in my analytic work for the 4th model, I determined some dynamics that were previously unknown from a prior work that had done a numerical investigation, but because of the numerical nature of their work, they did not determine all of the dynamics. And so my analytic approach was able to determine some additional dynamics that had previously been undiscovered. So that was a neat result. Okay, as we're continuing to move forward into the climate change motivated section, we'll turn our attention to the Integradifference equation models. So climate change is a powerful force in our modern world, and it's been shown that habitat ranges for different species are shifting polewards and or upwards in elevation. So the guiding question here is, can a population persist when its habitat is moving? And how do climate-driven range shifts impact the persistence of a specialist species, the parasitoid, that depends on another species, the host, for its survival? So to introduce Integradifference equations, this is a spatially explicit approach for discrete time models. So we're keeping that discrete time framework for these particular species. We're now adding continuous space, and in this particular system, I'm using one-dimensional space. Models do exist where you add a second spatial dimension, but that complicates the analysis. So we're starting with kind of the basic problem here. So these models take the form of looking at the population density of, say, the host at a spatial location X, and that will at time t+1, and that is determined by some stationary population dynamics that occur for the previous population, and then after those stationary dynamics occur, the population undergoes dispersal. So the individuals move from location Y to location X according to the kernel or the dispersal kernel here, and the individuals who end up at location X is the sum over all possible locations Y that contribute to location X in our one-dimensional spatial habitat, omega. One of the primary features of integradifference equations that makes them so interesting and useful is that there's a flexibility in what kernel you use to describe the dispersal of your population. And so you can incorporate a kernel that matches the observed ecological dispersal. For my purposes, I'll be using the Laplace kernel, which is leptokurtic, kind of describing the width of it, or the fatness of the tails. It's not the perfect match for population dispersal, but it is a more realistic than assuming a Gaussian dispersal. So it is used in modeling dispersal, and it also has some nice mathematical properties that make it a worthwhile kernel to use. So specifically, the integradifference equations that I'm looking at are those that have a moving habitat. So our one-dimensional habitat is now taking the form of a patch of length L that is moving rightwards at speed c. So the left edge of the patch is minus L/2 plus c times t. The right edge of this patch is L/2 plus ct. Our kernel, I'm also going to assume, is going to depend on the distance between the starting point y and the ending point x. So we have a convolution integral here. The Laplace kernel does this. And because we have the shifting spatial domain, that's simulating climate change. And it's been shown for single species that there's a critical speed, c star, beyond which the population cannot persist. So we're interested in what is that critical speed. As a bit of an illustration here, if we have some population that's distributed across space here, prior to thinking about climate change, we have this population distribution where the habitat is this, habitat patch is this range right here. Individuals inside the patch will reproduce, and then they disperse both inside and outside the patch. When we add climate change, that means that at each iteration or each generation, the patch is going to be shifting. So at this time step, our next time step, the population distribution will look like this, where our new habitat patch has shifted to the right. And in gray, I'm showing the previous population distribution. And then in black, we see that the population has shifted to the right with its habitat patch shifting. So kind of keep that idea of these shifting distributions as we're walking through this. For the Host-Parasitoid IDE system, again, coming back to the idea of discrete stages in the life cycle, well, the models that I'm looking at will have the host density dependence occurring first, followed by parasitism, followed by dispersal, which occurs in the adult stages for insects. So here are the equations. We have the density dependence occurring here, followed by survival from parasitism here, followed by dispersal, and then tallying up those dispersal sources at location X. And then here's the equation for parasitoid, the distribution of the parasitoid population. And I want to mention that in the following work that I've done, I assumed fractional recruitment and fractional parasitism to get a handle on the basic persistence question. When you add some of those exponential functions, you have the potential for things like pattern formation. And so to start in this work, I assumed the simplest case to give us a handle on that problem before adding additional crazy phenomena. So assuming fractional for both as we move forward. To, again, help illustrate what's going on with these different phases, if we think about location Y, a particular host, the host adults at location Y will lay eggs, the eggs will become larvae. The parasitism either occurs in the end of the larval stage or in the pupal stage. So hosts to pupae, those that survive become adults, and it's the host adults that disperse from location Y to location X. Sort of similarly, parasitoids at location Y will lay their eggs on the host. Those eggs become larvae, pupae into adults, and it's the adults who disperse from Y to X. and in the next generation, things occur again at that location. So demonstrating the sequential nature of those steps. Both kind of mathematically and ecologically, I'm going to be analyzing 3 cases. In the first case, I'm going to consider essentially the best possible scenario for the parasitoid. I'm going to assume that the host is going to be at its carrying capacity everywhere, so it's going to be on an infinite patch, and that the parasitoid is on a finite habitat patch moving at speed c, and that the host will be at its carrying capacity wherever the parasitoid encounters the host. In the second case, I remove that assumption about the host and instead look at the case where both the host and parasitoid are on the same habitat patch, and that's shifting right at speed c. In the third case, I'm gonna remove that biological assumption and suppose instead the host patch is larger inside, the parasitoid patch is smaller and exists inside the host patch, both moving at the same speed. And for simplicity, I'm assuming that the parasitoid patch is centered within the host patch. There's a lot of different scenarios you could model, but I'm specifically looking at the case where the parasitoid is centered within the host patch. So the question here is, we'd like to understand the basic persistence problem in the moving habitat for the host parasitoid system. What is that critical speed for coexistence? And how do these different types of models give us the ability to determine that, that critical speed for the parasitoid? For the first case, our host is at its carrying capacity 1 everywhere. So we really only have a single integradifference equation. This 1 can be plugged in everywhere. We see an n down here. So the system reduces down to a single equation. L is the patch length, C is the speed of the patch's movement, B is our clutch size parameter after non-dimensionalization. We have our dispersal kernel specified to be Laplace for the results that you'll see, though it could be done with other kernels. We have our per capita recruitment, and we have our parasitism functions. So that's our first model. In the second model, I'll have both the host population and the parasitoid population on that same patch of length L moving at speed c. Here, for simplicity of analysis, I've introduced the functions u and v, but that just encapsulates the competition and the parasitism, notationally simpler for analysis. And we'll see that we have a host kernel here and a parasitoid kernel here. To give you an illustration of what that looks like, let's take a look at a simulation. So I'll have the host here with space across this axis, parasitoid distribution on this axis, again space. And so each of these curves represents the distribution of the population at a particular point in time. So you see t equals 0, t equals 10, up to t equals 120. So we start off with no climate change, no habitat movement, and this is the distribution that we see for the host and parasitoid. Once the patch starts moving, the parasitoid population starts decreasing in size until it reaches a new steady state in the new moving habitat framework. The decrease in the parasitoid population allows the host population to experience a release, an ecological release, and so the host population actually increases again to some sort of steady state distribution on the moving habitat frame. If speed is too high, you will see the parasitoid population actually dying out, tinier and tinier little bumps until it's nothing, and the host will go to its single species dynamics in that scenario. Our third case is where the host patch and the parasitoid patch are different. The parasitoid patch is within the host patch, and again, assuming the parasitoid patch is centered, these are the governing equations, and I think everything else here is the same. But we get some interesting shapes when we look at these simulations. So again, space is on this axis, hosts here, parasitoids here. For this particular simulation, I'm assuming that the host is on a patch of length 2 and the parasitoid is on a patch of length 1. So we can see that in the kind of the region where the hosts and parasitoids overlap, the hosts are at a fairly low population size, but in their refuge regions, both to the left and to the right of that overlapping area, the hosts are able to be almost at their carrying capacity. Again, this thicker line is prior to habitat movement. Parasitoids have a little bit of a different shape than we saw previously. When we add climate change and start moving to the right, we see that the front part of the host's patch loses its effectiveness as a refuge, as the parasitoid is able to kind of invade into that space due to the movement of the patch. And so the host still has a refuge, but has lost some of its refuge habitat, and the parasitoid kind of has a peak as it's able to take advantage of some of that leading part of the patch. So we get to see some different shapes and some interesting features there with case 3. Okay, coming back to case 1, how are we actually going to determine the critical speed? For case 1, remember that it is a single integradifference equation, which has been studied by other authors. So following those traditional methods, we assume a traveling pulse and we shift to the moving habitat frame, which means that I now am looking at things in terms of an x hat and a y hat parameter. Our spatial integration is now over minus L over 2 to L over 2, and the C is showing up inside the kernel. This means that the kernel is now no longer symmetric, and that's going to be the source of the challenges in some of these problems later. But to proceed with the analysis, I linearize around extinction for the parasitoid. This means that if extinction is unstable, that means that persistence of the parasitoid population is stable. So we write an eigenvalue problem and use established numerical methods to determine the critical speed C, which is when the dominant eigenvalue of the system is equal to 1. With that setup, I can look at the critical speed and how that changes depending on what biological parameters we use. And so in this particular figure, I'm showing critical speed as we look at changing parasitoid average dispersal distance, and also looking at that clutch size parameter. Each of these curves is a different value of B, different clutch size parameter value. We see that as clutch size increases, the critical speed increases. Initially, for low values of parasitoid average dispersal distance, increasing the dispersal distance increases your critical speed. But at some point, if the population continues to increase the dispersal distance, it will actually over-disperse beyond that finite patch, and the critical speed decreases until the population goes extinct at all speeds. So that's one illustration of some biological insight we can get from doing models just with the case 1 scenario. I'll also be bringing back case 1 as we compare with cases 2 and 3. In the second case, we have that full IDE system of 2 integradifference equations. There they are for a quick reference. And the challenge here, once we have 2 species, is that I cannot linearize around 0, 0, because instability of 0, 0 does not correspond to coexistence because of that intermediate state where the host exists without the parasitoid. So 2 of the options that I chose to pursue, not to say that these are the only possibilities, but the 2 that I pursued in this work, were to first use simulations to determine the critical speed for a particular set of biological parameters. Quite computationally expensive to do this, but developing this option allows us to compare other approximations with what we're getting from the simulations. The second option that I pursued was separating the stationary population dynamics from the dispersal by looking at the average populations over the patch. So the bar representing the average over the patch. So these equations here, the N and the U, the P and the V, this represents those stationary population dynamics. And then summarizing this dispersal with this phi and n, which are between 0 and 1. Those are the percentage of the population that remains in the patch after dispersal. So that's the spatially implicit approach for this modeling process. So if we first take a look at using simulations to determine the critical speed, the main question here is based on the need to distinguish between cases where if you're near the critical speed but just below, you'll see that the parasitoid population will get much smaller, but it might persist at a very tiny value asymptotically. If you increase speed a little bit more beyond that, you might see tiny values initially, but asymptotically it dies out. So the question essentially is, how many iterations are we willing to do to determine if the parasitoid survives or not? And how do you actually determine between asymptotic survival at tiny, tiny values versus actual extinction? But we need to do that with finitely many iterations. So to investigate that question, I looked at fixing a set of biological parameters and running simulations at different habitat speeds, where I then calculated the maximum parasitoid population density after a fixed number of iterations. So each of the lines here corresponds to starting with 200 iterations. Each line is 200, 300, 400 iterations. So you see that initially for these lower speeds, we can't distinguish, there's no difference between the number of iterations, and we just see the maximum parasitoid population density as kind of is at a particular value independent of the number of iterations. The population density is decreasing until it drops off drastically. And so somewhere in this area is the speed for which the parasitoid population is going to die off asymptotically. If we zoom in around that drastic decrease, now we can kind of see the difference between the number of iterations. So this is after 200, 300, 400, 500, etc., down to 4,000 iterations. And so we can see that for this particular speed, there's a huge difference between the population after 200 iterations and after 4,000 iterations. If we move over here, there's essentially no difference between 200 and 4,000. So the question is, what threshold are we going to use to determine the critical speed? This dotted line here is 10 to the negative third. And the distinction that I made is that if, based on the fact that at this speed, and we see that there's a fairly large decrease from 200 to 4,000 iterations, whereas this one could end up asymptotically surviving. This is the point, I'm going to say that this is the case where it asymptotically goes extinct. This is the case where asymptotically persists at low values. And to determine that with a finite number of iterations, notice that this is the case where 1,000 iterations is below 10 to the negative third. So 1,000 iterations is this thicker line. And so that's the threshold that I'm going to be using for these simulations is if after 1,000 iterations, the maximum parasitoid population density is below 10 to the negative third, that's going to be my cutoff and that's going to be my critical speed. Because we can reasonably conclude that the population will drop off infinitely, will drop off in infinite time to 0. The methodology for the spatially implicit approximation case is obviously very different. With this model and approach to the system, I write down a difference equation system for the average populations, and we see this phi n and the phi p, which are the proportion of the population that remains in the patch after dispersal. Doing kind of standard analysis on the difference equation system, I was able to determine that the coexistence equilibrium is stable if this condition is met, where the the parasitoid parameters are here and the host parameters are on this side, and the critical speed is the speed for which this equation is met. I'll note at this point that these phi values here are based on both the choice of what kernel you're using, and then as a reminder, the kernel is evaluated at x plus c minus y. So the speed actually is hidden in these equations inside the phi function. So c determines these phis, and we're looking for the c for which this equality is satisfied. I, at this point, need to think about how to approximate the proportions of the populations that remain inside the patch after dispersal. And this is a very interesting mathematical question. Ultimately, it boils down to the dominant eigenvalue of an integral operator, defined here where we have the kernel and operating on some function phi, and we're looking for the dominant eigenvalue. The challenge is that the kernel is asymmetric due to the presence of C. And so some of the standard methods that work for symmetric kernels don't work, or are sloppy, I guess you could say, in their ability to estimate the dominant eigenvalue for an asymmetric kernel. One other approach is to take this, this kernel and discretize it, discretize this integral operators, excuse me, and to find the dominant eigenvalue of the resulting matrix. So there's a number of ways of doing that. Again, the challenge is we have an asymmetric matrix, and so can't use some of the nice results for symmetric matrices. In my work, I looked at a total of 6 different ways of approximating this phi n and phi p. The first 2 were developed for integradifference equations on a finite patch, but stationary, so no movement. And they do, they've been shown to have a broad applicability in that context, but don't do as well with approximations when you have that climate change, that asymmetry. But I'll compare those for completion. Next kind of category that I'm looking at is that of maximum and minimum row sums as estimates of bounds on the dominant eigenvalue. And there is a method that takes the kind of initial maximum and minimum row sum estimate and iterates to get better bounds on the dominant eigenvalue. And then the final 2 that I'm looking at are geometric symmetrization and iterated geometric symmetrization, which have been developed initially in the context of applied ecologically in the context of a single species on a moving habitat. And so looking at, can these tools also have value once we add the biotic interaction, and what does that look like? So looking at those 6 different methods. So let's take a look at how these different methods stack up and comparing our simulation critical speeds and our approximated critical speeds. So here I'm showing the critical speed where I've fixed all of the parameters except those that you're seeing on this axis. Black is the critical speed from the simulations. The blue is the critical speed from the method using geometric symmetrization. Green is from Reimer's modified average dispersal success, and orange is from average dispersal success. So primarily what I want to emphasize here is that geometric symmetrization is a lower bound on the critical speed, and that geometric symmetrization mirrors the qualitative behavior of the critical speed as biological parameters are varied, where we don't see that happening with either average dispersal success or Reimer's modification. So it's compared to both of these methods, geometric symmetrization is a better estimate of the critical speed, and it gives us this qualitative behavior that is important for understanding the systems. The next set of methods that I want to show are those including the row sums and the iterated row sums. So this purple line up here, drastically overestimating the critical speed, is from just the maximum row sum estimate. And the red curves are showing the maximum iterated row sum method and the minimum iterated row sum. Both of the curves shown here are after 2 iterations of the method. So the benefit with these is we do get both an upper and a lower bound. The upper bound is not particularly tight. Note that 2 iterations of this method, we're still not getting as tight of a lower bound as the geometric symmetrization, though at least with the lower bound, we are getting qualitative matching, but at a bit of a computational cost as well. The final comparison to show here is that of the geometric symmetrization in blue, and 1 iteration is what I'm showing here of iterated geometric symmetrization. So we do get a tighter bound when we use iterated geometric symmetrization. There is a computational cost, in particular for the Laplace kernel. I can run getting these critical speeds from the geometric symmetrization on a laptop very, very quickly, while the iterated geometric symmetrization takes longer because of increased complexity in the algorithm. But those 2 methods both give us nice lower bounds on the critical speed. So kind of to sum up those ideas, both geometric symmetrization and iterated geometric symmetrization have been shown in the methods that I compared to be the most useful approximations for the proportion of the population remaining inside the patch after dispersal. We get lower bounds, and ecologically, we have conservative estimates on the fastest climate speed that the parasitoid can survive. And again, ability to look at how this critical speed is impacted by changing biological parameters, that qualitative matching that we see. So I also want to talk about an upper bound. So we looked at lower bounds and determined the geometric symmetrization and iterated geometric symmetrization are kind of the answer in that area. And now I want to do a comparison of those upper bounds that we see. So that was the maximum row sums, iterated maximum row sums, and then case 1 is actually an upper bound because it was kind of the best scenario for the parasitoid. So when we look at how these different methods do, here I'm showing, again, biological parameters on this axis, critical speed here. Blue is geometric symmetrization. Just for comparison, the black is the results from simulations. Pink here is case 1. Green is Reimer, which isn't actually a true, it's not an upper bound, but I put it in these figures for comparison. And the red here is the maximum, the iterated maximum row sum method, 2 iterations, leaving off out of these pictures the maximum row sum by itself because it's such a high estimate that it doesn't show up on the figures. So what we see is that as the host net reproductive rate increases, case 1 is a really tight bound on the critical speed from the simulations. As we vary the host average dispersal distance in most ranges, we still see that this case 1 upper bound is the best of our upper bound options, though not for all values of the host dispersal distance. If I look at how B, the clutch size increases, the critical speed from case 1 is still a decent upper bound compared to our other options. But what I'm now going to sort of reveal, I guess, is that each of these cases that I've shown you have used a value of the parasitoid average dispersal distance that's right here. And so as we increase the parasitoid average dispersal distance, the case 1 estimate shoots up and becomes much less effective as an estimate of the critical speed. And so it's not, while it may be useful for small values of parasitoid average dispersal distance, it's not a useful upper bound in other cases. So kind of the conclusion I can draw from this upper bound investigation is that if the parasitoid average dispersal distance is sufficiently small, then case 1 is a decent upper bound for the critical speed for coexistence. But in general, if I make assumptions about the host population to reduce to the case 1 single IDE, I lose the ability to get a good estimate of the critical speed for the parasitoid persistence. So suggesting that it really is important to model the dynamics of both species to get a handle on persistence of the parasitoid species. So from kind of these different methods that we can use to determine the critical speed for the parasitoid, the geometric symmetrization is the method that kind of stands out. And so I can use that to test the impacts of the parameters on the critical speed to get some insights into the biological interactions. I won't include figures here, but just a few kind of things to summarize some of my results. The critical speed for parasitoid persistence increases monotonically as we increase the net reproductive rate for the host. However, there is an upper bound on how much increase there can be. And so continuing to increase R is no longer effective past a certain point. The critical speed for the parasitoid persistence also increases monotonically as we increase the non-dimensionalized clutch size. From what I can tell, there's no apparent upper bound on the critical speed as we continue to increase B. And both for the host and the parasitoid average dispersal distance, the critical speed initially increases as you increase the average dispersal distance, but ultimately decreases when the populations over disperse outside of making use of the population, of the habitat that's available to them. Now that we've kind of looked at that case 2, where we have the host and the parasitoid on the same habitat patch, I wanted to investigate what happens when the host and the parasitoid are on different habitat patches. And so I looked at what happens when the parasitoid patch is inside the host patch, and can we still use some of these techniques to get estimates of the critical speed. So I did do a similar process for the simulation method, determining a number of iterations to get the critical speed. And then I also looked at the spatially implicit method and writing down a difference equation system for the average populations. Because of the, there's essentially 3 regions in the hosts patch. There's the region at the back of the patch where there's no parasitoids. There's the region of the patch where the host and the parasitoid overlap, and your dynamics are given here. And then there's the region at the front where there's, again, no parasitoid. So our equation system is a bit more complicated here, and the trick was to look at how can I use geometric symmetrization, which worked so nicely for case 2, to approximate these 3 different coefficients accounting for those multiple regions. And then once I had that, being able to determine the stability criteria for the coexistence equilibrium, and from that determining the critical speed for parasitoid persistence. So geometric symmetrization ultimately did give us, again, a lower bound for the critical speed for the 3rd case. Again, it did mirror the behavior of the simulation critical speeds as we changed the biological parameters, so that was good to see. It was a looser bound on the, a looser lower bound on the critical speed than we saw in case 2. So something that I'd like to look into improving in the future. And then comparing case 3 and case 1, I saw that for smaller parasitoid dispersal distances with larger R0 and larger host patch length, case 1 was actually a closer estimate than case 3, geometric symmetrization, of the true parasitoid critical speed. And so case 1 was kind of a preferable approximate under certain conditions. So specifically to describe some of that, the blue curve here is the critical speed from the geometric symmetrization for case 3. The black, again, is the simulations. The pink is the case 1 approximation. And these figures I'm showing the host patch length. So as host patch length increases, case 1 becomes a better approximation for the critical speed. And then over here, showing kind of a different view on the same thing, if we look at the proportion of the host patch that is shared by the parasitoid, when the host patch is primarily just the host, we see that case 1 is a nice estimate, but as those two patches become closer to being the same, case 1 becomes less tight of an estimate on the critical speed from the simulations. So I've kind of thrown a lot with these 3 different cases and different methods to look at critical speed. Here are some of the takeaway messages from the work that I've done here. In general, it's important to model close biotic interactions to estimate critical speed. By making approximations to reduce the system to a single IDE, we lost the ability to get good estimates of the critical speed for the parasitoid persistence. On the other hand, when we, instead of explicitly modeling space, when I implicitly modeled space and looked at average populations, I was still able to get an answer to the basic persistence question for coexistence for those 2 species. And so space doesn't necessarily need to be modeled explicitly to answer this basic persistence question. In terms of geometric symmetrization, I've shown that it is a useful approximation for moving habitat models for interacting species. It is flexible enough to permit modifications to the habitat, as I did in case 3, making some different assumptions. The sort of negative on the geometric symmetrization side is that it is still an open question mathematically about how to rigorously show the relationship between the geometric symmetrization and the dominant eigenvalue of the original operator. And so not standing on incredibly solid mathematical footing, I guess you could say, but it's, as my advisor likes to say, it's kind of a case of the proof is in the pudding. I'm adding to a body of work that's saying that the geometric symmetrization method is useful, and maybe now we should tackle that why question. I'll kind of close this part by saying that the iterated geometric symmetrization method is useful to get tighter lower bounds if you're needing to get more accurate estimates of the actual critical speed for a particular population. So that's kind of the method that I would use for those types of questions. In concluding kind of the broad scope of what I've done, I've looked at host parasitoid models, both on a non-spatial scenario of looking at difference equations, have looked at the impacts of the scramble competition and the contest competition, what happens when we assume that the population, the parasitoids, are searching randomly versus aggregating, and how do those different biological phenomena impact the types of population dynamics that we might see, and looking at how those individual components impact the dynamics. I've also looked at the moving habitat case once we introduced climate change, and showed that it is important to model those biotic interactions to look at the persistence question of the parasitoid, those interacting species, and compared what happens when we look at space implicitly instead of explicitly representing space, and the types of questions that we can answer when we make those simplifications to our models. So yeah, I would just like to say thanks to Dr. Mark Cott, who's been my advisor for this process, for his guidance and his insight. Thank you also to my committee members for their support, their insight and encouragement. I also really want to extend a hearty thank you to the Applied Math department as a whole. The community here has been phenomenal, from my cohort to other students, the staff, the faculty. It's been just an incredible place, largely because of the people who have been part of this journey. And also many thanks to family and friends and my church groups for their support and encouragement throughout this journey. Thank you.
Okay, let's give a round of applause to Kelsey for her talk first. Okay, sorry you can't hear all the clapping. And then let me just remind everybody how this kind of works at this point. At this point, what I'll do is I'll briefly throw this open to general questions from the general audience. Okay, anybody can ask a question, and I'll let Kelsey field them. And then after she's answered several of these questions, then what we'll do is we'll excuse the general audience and the committee will remain. We'll actually take a break at that point so that everybody can grab a glass of water and so forth and catch their breath, okay? And then we'll go into closed session, okay? So thank you. First of so I'm going to throw the floor open to general questions, but also let me just take this opportunity, since I will excuse you at some point, let me take this general opportunity now to thank everybody who showed up. I think it's really wonderful. It looks like there was very, very good attendance, and I can't thank you all enough for showing up. Thank you. Questions?
I have a question.
Go for it, Catherine.
So today there was an article that UW Today sent an email about parasites and someone studying, looking at how they're changing with climate change. And so I read that to be prepared for this. But one thing that they mentioned is that there's sort of two trajectories that they see happening. One is parasites going extinct, which I seems like you were really addressing with your models. And another one was looking at massive outbreaks of parasites. And I guess I was curious if you could see adapting these models in a way that might be able to show that trajectory.
Yeah, so they don't send me UW Today anymore, so I haven't seen the existence of this article, but I'll do my best to answer your question. I know that in some of the work that I've looked at, in addition to looking at just the critical speed for parasitoid survival, I have seen cases when I've done simulations that give me more of an insight to this, like kind of the spatial distribution of parasitoids, where they actually, the simulation that I showed early on shows the parasitoid peak decreasing. So the parasitoid peak decreasing here, but at other speeds, the parasitoid peak actually increases when we add climate change. The total population size decreased, but the peak increased. And so my model does actually predict what you could consider outbreaks of the parasitoid where the density is higher at particular spatial areas. So yes, that is something that my model has the ability to show. The challenge is, yeah, I think kind of determining maybe the ranges of the climate speed for which that happens, because of that question kind of being a spatially explicit question. At the moment, I would have to address that with the simulations rather than the spatially implicit difference equations, but that is something that would be interesting to explore.
Cool, thanks, Kelsey.
Thanks, Catherine. Any other questions? Okay, so if let's see if I've got, I'm not sure I see everybody. I'll try to show everybody. So if there are no other questions, okay, do you see anybody, Kelsey? I'm not sure I can see everybody.
I'm scrolling through. Everybody's keeping their faces covered and hiding.
Okay, So if there are no other questions from the general audience, let's take this opportunity to once again, let me take this opportunity to thank everybody, and let's all give Kelsey another round of applause.